So when scientists do experiments, they have to rely on reliability. Your information must be reliable. The most reliable resource for a scientist would be a scientific journal, with the lowest being articles on the internet. Why do you think that articles on the internet would have the lowest reliability? You can write your answer down below. Scientific journals, they undergo peer review. But what is peer review? Peer review is when I write a paper and I give it to somebody else and the other person reads over it to see, okay, maybe that does make sense. Or what if it doesn't? They can perform the experiment themselves. If they don't get the same results, then maybe it's not a reliable source. This is why it's important when we're recording our data in a lab book that everything matches up. We want to write down exactly everything we do. That way the next person coming along will be able to match it up exactly. So when you do get stuff on the internet, there are certain websites that may be okay. The highest would be obviously a .edu, which is an education site, then a .gov, which is the government. Then we have .org, which is not really okay, but it is an organization, so maybe sometimes. And then the least is .com. When you're doing something in my class, never, ever, ever quote from a .com website, and never, ever, ever quote from Wikipedia. Those will be marked down highly. A scientific question is one that can be tested. You'd be surprised at the type of questions that cannot be tested. For example, you cannot test animals in the wild. Why? Because you never have a set standards for them outside. Now, when they're in the zoo, yeah, you can control that, but you can't control what's going to happen in the wild. So we also can't test what your favorite color is. I don't have a clue what your favorite color is, and as far as I'm concerned, it could change today, and 2 minutes later, it's something completely different. So a good scientific question is one that can be tested. We can obviously test which type of fruit gives me the best example for DNA. We can test which paper quality works the best in this printer. We can test which glue holds the best stick. Those are things that we can test. So you need to list 3 good scientific questions and then 3 poor scientific questions. When doing an experiment, there are multiple things that have to play into it. Not everything is exactly the same every time you do it. First off, what is a variable? A variable is a fact that can affect the outcome of an experiment. So this is something that can change what's going to happen. There are 2 types of these. You have an independent variable, and it is the one that changes, and the dependent variable, which changes in response to the independent variable. So what are some examples of an independent and dependent variable? So the independent variable, again, is one that changes. So let's say we have 2 flowers in our classroom, and they got the same type of soil and everything. So what could be the independent variable? Let's say that the independent variable is the amount of water that each one receives. So plant A, we're going to say, receives 1 cup of water a day, and plant B receives a half a cup of water a day. So that's the independent variable. Now what changes? That would be the dependent variable. The dependent variable in this case might be the height of the plant. So that's an example of our experiment with the independent and dependent variables listed. There are 2 different groups as well in an experiment. You have your experimental group, which is the group for which the independent variable changed. So our experimental group was, let's say, plant B. And the control group is the group to which the experimental group is compared to. So that would be plant A. So we're comparing plant B to plant A. Plant A is going to be the normal rolling group. So with an experiment, you can collect 2 types of data. You can have qualitative data or quantitative data. Qualitative data is stuff that's in words. So something that's in words might be blue shirt, brown hair, green eyes. That's all qualitative data. Quantitative data, that one's pretty easy to remember. It's in numbers. Think of it. You're in math, quantities. It's all math. So those are numbers. So 5 centimeters tall, 6 inches long. Those are your quantitative data. Then we have a theory. A theory does not become a law, nor does a law become a theory. This is something you will need to memorize. It is very, very important, and I promise you, you will be tested on it. A theory is a well-supported explanation. So this is why it happens. Then we have a law, which is a well-supported description. This is actually what happens. Make sure that you understand the difference between those two. The next activity we're going to do is the characteristics of life foldable, and this actually introduces the whole concept of biology. This is something you will need to pick up from me in the classroom. If you're doing this for a review, just find it in your folder and review it. So microscopes. What is a microscope? A microscope is obviously something that you use for something that you cannot see with the naked eye. There are different types of microscopes. We will go into those. I doubt we will do a lab as of now with a microscope, but probably later on. Most of you have used a microscope before, therefore it's not really necessary to do it now. And I want to cover this unit pretty fast, but if I realize it's something we need to hit, I will hit it then. Make sure that you understand the basics of the microscope. The main thing I believe you will be asked is obviously on the different types of microscopes. There might be a few questions as to what piece goes where. I will most likely quiz you on this at some point or another. It will be an identification thing as well as just some basic facts as to what is a microscope. There are two types of microscopes that we're going to learn about. First one is light, and then we have the electron. The light microscope is just something that you use in the classroom. It's very basic. It includes your dissecting and your compound. This type of microscope uses lenses and light to magnify objects, and it's what is obviously used in most science labs. The electron microscopes are pretty cool. They're pretty in-depth. Don't really see these very often. Luckily, me and some STEM students got to see them this summer. I personally had never seen them before then. This includes your scanning electron microscope. That's your SEM. And then your transmission electron microscope. That's your TEM. Those are two things that you just need to memorize, because I will definitely ask you what TEM stands for and SEM. So TEM, the transmission electron microscope, transmits electrons through a thin slice. The key thing here is that it makes a 2D image, where the scanning electron microscope, SEM, scans the surface. With this one, the computer forms a 3D image. The TEM microscope and the SEM are both done in vacuum-sealed rooms, because you definitely do not want to lose your particles. When they're using these, they're using particles that could just disappear every breath you take. Now we're going to cover the basic parts of a microscope. These, again, are important to know if you were to ever use a microscope, especially if you're in my biotech class. You will be using these and need to know exactly what these are. I know the definitions are posted later, but I do want to give you a brief summary as to my definition for what each one of these are. So first we have the eyepiece. The eyepiece is obviously what you look through to see what is on the stage. The stage is what holds your slide, which is glass, and that is what your specimen is on. Stage clips are what hold the slide in place. Your objective lens are what you change to see different depths of the object. For example, when you start at the 4, you're seeing right above the object. It's not a very in-depth picture. Show you a wide range. That's the 4 times. Then you move up to the 10 times, which is the medium power. Get a little bit closer. Then you reach the 40 times. When you reach the 40 times, you absolutely do not touch the coarse adjustment knob. The coarse adjustment knob is what moves the stage up and down. If you are to touch the coarse adjustment knob when you have the objective lens in 40 times, you risk, first off, losing your specimen, as well as you risk breaking the slide. So all in all, it's just a bad idea. The fine adjustment knob is something that you can touch, and that kind of focuses your object into what you want to see. The light source, obviously that's where the light comes from. The base is what the microscope, the bottom of the microscope. If you're ever carrying a microscope, you always hold it by the arm and by the base, never by anything else. The nose piece, that's what the objective lenses are connected to. That's what you rotate to change them. The diaphragm is what lets the light through. And that pretty much sums up all of your pieces to a microscope. Make sure that you have all these definitions filled in. There is a picture there as a key in case you missed any of the labeling earlier. This sums up the microscope, and that pretty much sums up the unit for Unit 1. We will have a test pretty soon. Make sure you study. This is what all will be covered. Let me just note at the end of your packet there will be some questions. You need to make sure that you answer those questions before you turn in your notes. That is part of the requirement. These questions are questions that I've found from EOC practice stuff, so they are important. They will be on your EOC, or there will be similar ones like it. Make sure you study these. I'm pretty good about giving a study guide, so plan on there being a study guide for this test. In the classroom, I'll go over these questions. Again, if you missed it, sorry. Get it from somebody. If you have any questions, make sure you come see me before the test. I do not advise it after it. Thank you.